What reductionism allows you to do is to take a complex problem and focus on one component of it and try to understand it in some detail. And sometimes you can just do it by focusing on one component. Other times it requires selecting a particular biological system, if you're working in biology, in which that component is prominent or easy to study. And that allows you to study in depth a problem that would be hard to do if you looked at it in all of its complexity. For me, the reductionist approach was really very profitable and not something that I really thought a lot about before. I originally went to medical school with the idea of becoming a psychoanalyst. I didn't have a strong biological background at all. And then in my senior year at medical school, there was a 5-month elective period in which you could do whatever you wanted to. And I thought that even a psychoanalyst should know something about the brain. So I took an elective in brain science. There were very few people doing brain science in those days, but Columbia had an outstanding person, Harry Grunfest, and I worked in his lab, and I worked with one of his associates, Don Purpura, and had absolutely spectacular experience. I mean, actually doing science is so different than reading about it. There's a sensual pleasure of doing the experiments. There's the fun of thinking about it, coming up with a new finding. I just thought it was marvelous. And Denise and I had just decided to get married, and we had dinner one night. And I remember saying to her, you know, I could see doing this for the rest of my life, but it's completely unrealistic. You don't have any money, and I don't have any money. You know, we want to have kids. I should really go into practice. And she banged on the table and she said, money is of no significance. I should tell you, sotto voce, that in the subsequent 60 years, she's not repeated those words very often, but she did say them that night, and it made a big impact on me. So I decided to go ahead with science. And when I got out of medical school, which was in 1956, physicians were being drafted in the service for 2 years. But if you qualified for the National Institute of Health, that was served as an alternative to draft duty. So he nominated me, Grunfest, to the NIH and National Institute of Health, and I was selected. So I spent 3 years there. I spent the next 3 years doing brain biology. And I asked myself, what's the central problem in psychoanalysis? And I thought, memory storage. You know, we are who we are because of what we learn and what we remember. And psychoanalysis is designed to allow you to relive earlier experiences, earlier memories in a protected environment. Brenda Milner had just shown that the hippocampus in the mammalian brain is central for memory storage. And I had mastered in Grunfest's lab how to put electrodes into single cells. This is something, you know, many people can do now, but when he taught it to me, that relatively few people could do it. In fact, the whole population doing brain science was very small. So my colleague Walden Spencer, whom I recruited to join me on this, and I were the first people in the world to record from single cells in the hippocampus. We were euphoric. The people around us thought it was fantastic. You know, 2 incompetent people coming to the NIH, and the intellectual environment is so extraordinary, it brings them up to this high level. So we studied these single cells to characterize their properties, and we saw after a while they were not dramatically different from the other 2 or 3 groups of cells that have been described. So clearly learning doesn't reside just in the properties of cells. It should have been obvious at the beginning. It resides in how a behavior is modified by learning, how something changes in the neural network. And we had no idea what the sensory information coming into the hippocampus was. We tried various things, nothing worked. Turns out that space is very important. It's a complex modality. So I realized one needed to take a very different approach, a reductionist approach. Rather than going to the most complex example, human memory, take a very simple. Pavlov Thorndike just studied simple reflex behaviors and how they were modified by learning. So I began to look for an animal that had simple reflex behaviors, and it was advantageous from a nervous system point of view. And that's what led me to aplysia. It's an animal that has only 20,000 nerve cells compared to the 100 millions of nerve cells in your brain. It only has 20,000. And each of them is gigantic. Aplysia has the largest nerve cells in the animal kingdom. And you can easily put an electrode into any of these cells and be there for 24 hours if you wanted to. And the cells are not only large, they're uniquely identifiable. So you can return to the same cell in every animal of the species. So not only that, but it turns out that one of the most interesting laboratories, only 2 laboratories working on it, was in Paris. My wife is a Parisian chauvinist, so we went to Paris. The alternative was Marseille. Another person was working on Angelika Vanitaki-Charlesonitis worked in Marseille. And Denise said, going to Marseille is like going to Albany. We should go to Paris. So we went to Paris. We had a great year. And I started to work out a very simple behavior in the animal, a withdrawal reflex, like the withdrawal of a hand from a hot object. And I showed it could be modified by different forms of learning. And in each case, the learning involved a change in the strength of synaptic connections. So nerve cells connect with each other at junctions called synapses. And I found that those synapses are not fixed, but they're plastic. And they're modified by learning. Certain kinds of learning causes an increase in strength. Learn fear, classical conditioning. Some, like habituation, learning to forget about something, leads to a decrease in synaptic strength. Later I found there's a short-term memory and a long-term memory. A short-term memory is a change in functional connections that lasts from minutes to hours. And long-term memory is actually an anatomical change, a growth of new synaptic connections. So I really learned a great deal about this. And then in about 1980, I started a second front. I returned to the hippocampus. By that time, when I had a much better understanding. And it turned out that the rules there are pretty much the same here, except when it's looking at more complex behavior.